So at this part we actually get to start with something that's alive now finally. We're done with the chemistry section. And so it's looking at the cell. We spend the rest of the time looking at things with cells in this class in there. So your cell is the smallest living unit. And when we look at where the size is for something, you look at 0.1 nanometer is where atoms are. Then you get to where most bacteria in there are going to be 1 micron to 10 microns. Most plant and animal cells are 10 to 100 microns in there. The human egg is going to be that really large single cell. That's the largest human cell, is the human egg. It is almost big enough to be seen with the naked eye. Not quite, I guess. You know, it's one of those things, if you've got really good vision, you might be able to see it in there. And then you go up to the height of humans here and other things. I think we're a little off saying we have, oh, I guess they drew the line here. The human that's 10 meters tall. But so when you study cells, they're too small to be seen with the naked eye, so we use the microscope, which you got to start playing with yesterday. The light microscope is what we use in here, and it allows visible light to pass through the specimen, and then it goes through glass lenses that are going to do the magnification. With the light microscope, you can magnify up to about 1,000 times. So the magnification is the ratio of the object size to its real size. It's how many times you see the size, and so it's written with a number and then an X after it. The resolution is the clarity of the image. It's the minimum distance apart things need to be to still be able to be distinguished as 2 points. So sometimes if they are too close together, if it's closer than what the resolution of that microscope will allow, it will look like 1 point together in there. With the light microscope, it's about 2 microns in there, or 200 nanometers. And what limits it is the shortest wavelength of the light used. And that's why with the light microscope, you're never going to get any better than that, because it's the size of the waves of light in there. That's not going to change. The reason you can see better with the electron microscope, though, is because those wavelengths are smaller. So the contrast helps to accentuate the difference in the parts, and that's part of the reason we stain them. We also stain them because they are so small and so thin that the light can pass right through them, making it really hard to see them. So your cell walls were first seen by Robert Hooke in 1665. Anton von Leeuwenhoek was a lens maker that he made the first microscope. The first microscope was actually just like a magnifying glass that was a single lens in there. And he actually called them beasties, is the things what he called what he saw in there, the cells. Most of the organelles, which are going to be your membrane-bound compartments in a cell, are going to be too small to see, at least with the light microscope. So with the electron microscope, this was introduced in the 1950s, and it focuses the beam of electrons through the specimen or onto the surface. Its theoretical resolution is 0.0002 nanometers, but in practice it's still about 2 nanometers in there. So your resolution is also going to be limited by the other equipment on there. It will reveal more, but the process that you have to go through to get things ready to view them with the light microscope is a lot more intensive in there, and it's going to kill the cells, so you don't have the option of looking at anything alive in there. Light microscopes are going to be expensive, so they're not readily available to most people. A research lab, university, yes, that's one thing. Clover Park, TCC, any of those? No. Not going to happen in here. Honestly, if they brought one, none of us know how to use it, because even at the UW where they do have them, they didn't allow us to touch them. And then the stains for them, it's going to involve using heavy metals, so they're very toxic stains in there, so you have to be much more careful handling things, handling the stains. And they also have to be viewed in a vacuum. So it will allow you to see the cellular anatomy, though. The scanning electron microscope is going to show you the surface And then the transmission electron microscope will show you the internal structure. So cytology is just the study of cells. So this is showing the scanning electron microscope, showing cilia here, that you can see the surface of them. And so they're artificially colored this way. And then the transmission, it shows the slice right through the middle of them. And then these images over here just show what different types of microscope images would look like on there. What you're going to be used to is bright field stained is what we have access to here. So when you do cell fractionation, this takes cells apart and it's going to separate the major organelles and subcellular structures with a centrifuge. So basically you just mix them up. Do you need the key, Harold? And it's kind of like when you centrifuge blood where you have the different types of cells separate into different layers. You can do that with the cell components in there, and it just allows you to study them. Thank you. So when we compare the prokaryotes and eukaryotes, the big difference is the eukaryotes are going to have internal membrane bound compartments, where the prokaryotes do not in there. Both of them are going to have a plasma membrane, which acts as a selective barrier around the outside of the cells. And so this is showing an image of where the outside of the cell is. Then you've got the inside. This is an illustration that shows how you've got the phospholipid bilayer that makes up your plasma membrane. And then you've got these proteins embedded in there that are going to span all the way across the membrane. Inside you've got the cytosol, which is the semi-fluid-like substance that's going to contain the cellular components. There's a couple of terms that sound alike there, cytosol and cytoplasm. Cytoplasm is going to be the inside fluid-like substance with the components. The cytosol is just the fluid-like substance. So cytoplasm is the parts in the juice, cytosol is just the juice. So some common organelles that you're going to see in all cells. Chromosomes aren't really an organelle, but they got lumped into this category. They're going to carry genes, and then ribosomes. So you'll have that across the board in all the cells. The DNA is different in eukaryotic and prokaryotic cells. The eukaryotic cells are going to have most of their DNA in the nucleus, and then it gets surrounded by a double membrane. Prokaryotic cells don't have a nucleus, so instead they have all of their genetic information concentrated in a nucleoid region in there. So there's just no membrane around it, they just cluster it all together. So that's what you see there is your nucleoid region in there. It's just clustered in there, but nothing around it. So the cytoplasm is the inside of the cell area between the plasma membrane and the nucleus. So if you don't have a nucleus, it's just the whole area in between the plasma membrane in there. The size of the cells, eukaryotic cells are generally much larger than prokaryotic cells. Your largest prokaryotic cell is about the same size as your smallest eukaryotic cell in there. A lot of the cells, their size is going to be limited by their metabolic requirements. One of the important things that we look at in there is the ratio of the plasma membrane surface area to volume in there. The more membrane you have, the more stuff can get in and out of it, and the membrane does a lot of other things for the cell. It's going to control a lot of other reactions. So the bigger you get, the more volume you have for that membrane to accommodate. The membrane doesn't grow at the same rate that the inside of the cell does. The volume's going to increase much quicker in there. So this is showing it here where you do a comparison of the surface area in there being 6, 150, and 750 in there. So if you have one small cell here, your surface area is 6. If you have a big cell, it's 150. But if you make that big cell a whole bunch of small cells instead, now your surface area goes up to 750 in there. So it's much more efficient to have multiple small cells than one big cell. And when you look at things in the animal world, bigger animals don't have bigger cells. They have more cells in there. As you grow from being an infant, you don't have bigger cells. You grow more cells in there is what we see in the world. So those membranes, they help to separate things into the organelles. They'll facilitate and participate in metabolic functions in there. So they're involved with a lot of those chemical reactions, which is why we need to have more membrane space. So your genetic instructions, most of them are going to be in the nucleus, in a eukaryotic cell in there. It's going to have most of the genes. You do have a few that are outside. Mitochondrial DNA is outside of the nucleus in there. The nuclear envelope is going to enclose the nucleus. It's a double membrane, just like the lipid bilayer is, with its associated proteins in there, and then it will have pores that actually will perforate all the way across the membrane. So it's two membrane layers around the nucleus. The pore complex is going to have an inner and outer membrane, and it's going to be continuous at the lip of the pore. So when you've got your membrane here, this is your membrane and this is your pore. This is all continuous that makes this lip of the pore in there. It's not going to be separated and have a hole in it in there. And this is going to help to regulate the entry and exit of most proteins and RNAs. So you don't want to have things just randomly going in and out. This will help to control and say who gets in and who doesn't in there. Your nuclear lamina is a net-like of protein filaments that line the nuclear side of the envelope, and it helps to maintain its shape. So it's kind of like having a mesh of little fibers under here that are going to help to hold the shape of it better. And that's similar to what you have actually with your skin. You've got fibers underneath your skin that help hold things that are underneath in place as well. If you look at where you see veins on the back of your hands, they stay there because there is a membrane over a layer of fibers in there, and the fibers are all around those veins so that you can't take and say, you know what, I'm going to scoot them over to this side today. Doesn't work that way. Your nuclear matrix, this is going to be the fibers that run throughout the nuclear interior. So you've got these that are up closer, and then you have some that are going to continue all the way down afterwards. They're just not going to be as thick throughout the interior in there. Your chromosomes are going to be the organized form of DNA. When you're going to form these as in cell division, the chromosomes are those ones that we think of looking like the little X's. And that is the only time you have chromosomes is during cell division. The rest of the time you're going to have chromatin, which is going to be a complex of proteins in the DNA and the loose diffuse DNA. So what you're going to see your mass looking like most of the time is like a ball of threads in there. And although it looks like it's random in that pattern, it's not. We also have the nucleolus in there. If you've got multiple of them, they're called nucleoli. This will be in cells when they're not dividing. And this is going to help to synthesize the rRNA that you use to make proteins and make your ribosomes. And the ribosome has a large and small ribosomal subunit in there. So being that it's too big to get out a whole ribosome, That's part of the reason for having subunits, is it's kind of like saying, well, you need to move your house, we got to break it into pieces to move it. You're just breaking the ribosome into pieces to get it out the door. And then it also helps to regulate cell division. Another thing that has to go through these pores is your mRNA, which you'll make in the nucleus. Your DNA is going to stay in the nucleus, and it's your instructions to make proteins, but you're going to actually make the proteins outside the nucleus. And so if you have to have it made outside and the instructions are on the inside, you have to have a way of getting those instructions to the outside. That's what the mRNA, M is for messenger in there. It's going to be a copy of the DNA that's made and sent out to the cytoplasm. And then the ribosomes will translate it. So your ribosomes are the little protein factories in there. They are going to translate the DNA. And they are made up of a complex of ribosomal RNA and proteins in there. RNA also has a lot of similar properties to its structure that allow it to act like an enzyme in there and also act similar to a protein in the shape in there. And so you'll have numerous of these in cells that are going to do a high amount of protein synthesis. So can you think of some place in your body you might have cells that have a lot of RNA or ribosomes? So let's say, take your liver for example. It's got to make a lot of enzymes to digest things. Enzymes are made of proteins. You would have a large number of ribosomes in those cells. Some of the ribosomes are going to float around freely and some are going to be bound. The free ribosomes get suspended in the cytosol and they usually function in the cytosol. One of the things they do is they help to catalyze the first steps in sugar breakdown. The bound ribosomes are going to be attached to the ER, which is your endoplasmic reticulum. They'll also get attached to the nuclear envelope. And they get inserted into the membranes, and they'll help with the packaging within the organelles and help with exporting the secretion in there. So this is showing here where you've got free ribosomes versus bound ribosomes that are attached to a membrane. This is showing you have a large and a small subunit of the ribosome in there. So the endomembrane system is the membranes inside that are going to carry out various tasks. This includes protein synthesis and transport. They're involved with metabolism, moving lipids, helping to detoxify things. And the various membranes are not going to be identical in structure or function. So they'll all have different structures and do different things. When you have a sac full of a substance that has a membrane around it, we call it a vesicle in there. So you may have heard a term used like vesicles, little bubbles on the skin. Skin is a membrane around a fluid in there. That's all it is on the inside, just smaller vesicle in there. So your endoplasmic reticulum is really extensive. It's about half of the total membranes in the eukaryotes. Lots of tubes and sacs in there. Inside of the membrane tubes in there, you've got the cisternal space. And it's just separated from the cytosol in there, but it is continuous with the nuclear envelope. So if you look at it like this, that you've got a cell, and then you've got your nuclear envelope here. It's a double membrane around it. Try again. Your endoplasmic reticulum is continuous with it. So there's actually that connection in there. So your smooth ER doesn't have surface ribosomes on it. It will tend to be involved with more fatty things. So when you think about it, a membrane is made of fats in there. And if you think smooth ER is a membrane in there, think more of the fats that go with it. So it helps to make lipids, steroids, phospholipids in there. It helps in the detoxification of drugs in the liver. What it does is it attaches an alcohol group on there, or a hydroxide, to help to increase the solubility so that you can flush it out better. So one of the ways you get rid of things in your body is through urination, and when you look at your urine, hopefully you don't see stuff floating on it when it comes out. It's dissolved in there. And that's what is going to help to dissolve it in your body so that it can be dissolved in your urine and exit that way, or through sweat, other ways like that. Proliferation of the smooth ER can actually help to increase your tolerance to certain drugs. So when sometimes people will take a drug for a while, they have to take more and more of it to get the same effect, because the body's looking, any drug is basically a poison you're taking into your body for a desired effect in there. And the body gets, in a lot of cases, better and better at breaking it down. It becomes more and more efficient at it. And this is one of the things that allows it to become more efficient, is it says, oh, okay, I need to make this enzyme. And it looks at it after a few days and it says, well, apparently this job isn't going away. I need help. Let's make some more smooth ER so we can make more of this enzyme and do it more quickly in there. So it will also help to store calcium ions. This particular thing happens in muscle cells in there, and in muscle cells we call the endoplasmic reticulum the sarcoplasmic reticulum. So your rough ER, this is going to have ribosomes on the surface, and so ribosomes we know make proteins. So you can look at this and say, all right, more protein related things in this. So where you're going to do protein secretion, like making insulin, your glycoproteins, which are protein and carbohydrate, will be more involved with the rough ER. It makes the secretory proteins, and then it keeps them separated from the cytosol. So it can make the protein and use some of that membrane from the endoplasmic reticulum to wrap it up and keep it from interacting with the rest of the cell. And then it can also help to transport it through that membrane in there. Also, it gets involved with membrane synthesis, where it can add proteins and phospholipids to its own membrane in there to help make more membranes for the cell. So this is just showing where you've got the lumen on the inside, some smooth ER, some rough ER. And if you look really close, it kind of looks like one of those mazes where you can find the start and follow the line through. So the Golgi apparatus is a series of flattened membrane sacs, and they call the sac cisternae. What it tends to look like is a stack of pancakes in most illustrations of cells in there. And what it's going to help to do is modify and store proteins and get them ready to send to other destinations. Sometimes you may make a protein and it may have a short lifespan in there. It may be dangerous. There may be various other reasons that you're going to make most of a protein, but you don't want it activated until the last minute. The Golgi apparatus is a way of kind of controlling that in there so that you can get most of the job done and just turn it on at the last minute when you need to. So these are going to be extensive and secretory cells, and they have a definite polarity in there. They've got a cis and a trans face. So they've got a front end and a back end in there. And things are going to move through in a specific direction in there. So the cis face is going to receive the vesicles of the substance comes in. They go through and they exit on the trans side in there. And they get modified in transit along the way. So it helps to modify the carbohydrates that you find in glycoproteins. Helps to alter membrane phospholipids. can be used to make certain macromolecules in the cell, make a lot of polysaccharides in there. So the cisternal maturation model basically says it comes in the cis phase, goes out the trans phase, modified along the way in there. And so this just shows it coming in. A lot of times it's going to get marked with a molecular ID tag so that it tells it where to go afterwards. So the lysosomes, these are a membrane-bound sac of hydrolytic enzymes in there. So hydrolytic, when you look at the name of these things, a lot of times the names are overwhelming, but if you look at that hydrolytic, lysine, hydro with water, it means it's going to help use water to split something in there. It will be used to help digest macromolecules in plant cells. So proteins of the inner surface are going to protect the lysosomal membrane. So the reason it doesn't destroy itself is it's got special proteins lining the inside of it. So it's kind of like when you drink something out of a can. It's got a lining on the inside of it so that it doesn't interact with the can. So when we do phagocytosis, you can have a food vacuole or a smaller organism or something else that you take into the cell. It's going to fuse with the lysosome, release the enzymes in there, and then the enzymes digest it and breaks it down into the monomers that you use. So autophagy is when the cell uses hydrolytic enzymes to recycle its own materials. So it's just a way of digesting things within the cell to reuse the supplies. So an example of a disease where something goes wrong with this is Tay-Sachs disease, where the person is missing a lipid digesting enzyme or it's inactive in the brain, and it allows some of the lipids in there to start to accumulate and cause problems. It's actually a fairly rare disorder. So vacuoles, some examples of these, we've got food vacuoles. These are formed by phagocytosis of food. So when there is food on the outside, the cell engulfs it and puts it in a package, and we just call it a vacuole at that point. Contractile vacuoles are going to pump out excess water out of the cell. So they will just collect water from the inside and push it out. You'll see this in some of the freshwater protists. When you have something that is a cell living in fresh water, it's more concentrated inside the cell than it is outside the cell. There's more solids inside the cell. Water is naturally going to want to come in there to try and dilute the inside of the cell. But if that keeps happening, it's going to explode. This is how they prevent themselves from exploding. So if you think about it, certain fish, their cells have to have the ability to do this and be able to go back and forth. So if you're a salmon and you go back and forth between freshwater and seawater in there, you have to be able to adapt to both mechanisms. The central vacuole, this is in plants, and it's usually really big in plants. It's a coalescence of smaller vacuoles that carry out hydrolysis. It'll hold extra ions in there, helps to dispose metabolic byproducts, can have pigments in there, some poisonous compounds in there. So then we get to the mitochondria and chloroplasts. This is where cells are going to make energy. So that's why these are my favorite organelles, because more energy from them. Mitochondria is where you're going to do cellular respiration. That's what animal cells are going to do. The chloroplasts are going to be where you do photosynthesis. And that will be in some algae and plant cells in there. It's enclosed by a membrane, but it is not part of the endomembrane system in there. They're separate. The membranes get made by the free ribosomes, and inside they contain a small amount of DNA. So they are semi-autonomous. They can grow and reproduce within the cell. So if you need more mitochondria in your cell, in there, they can grow and reproduce, and you can have more of them produced within the cell. The place where you're going to have a lot of them is in a muscle, especially your endurance type muscle fibers. So when you first start exercising, one of the things that's going to happen is they are going to grow and reproduce themselves in there to help you adapt to exercise in there. So the other thing with them having that small amount of DNA, what they know, there's this thing called the Endosymbiont Theory, where they look and say that, okay, we've figured out how the eukaryotic cells have arose. We had prokaryotic cells beforehand, and you had a prokaryotic cell that ended up engulfing an oxygen-using, non-photosynthetic prokaryotic cell here. That cell that went in ended up becoming the mitochondria, and it lost the ability to survive on its own. So that's why it has its own DNA in there, is it actually used to be, a long time ago, an independent cell. Now, this doesn't happen individually in your own body. They start out as eukaryotic cells, but this is how we formed first eukaryotic cells. And then some of them took on photosynthetic prokaryotes that turned into chloroplasts in there. So the peroxisomes, these are another organelle. They're an oxidative organelle that's not part of the endomembrane system. So mitochondria are in nearly all eukaryotic cells. You've got a double membrane in there with phospholipid bilayer. So that double membrane also helps to suggest that it was a prokaryote that was engulfed in there. Here you can see you've got the membrane around the outside, and then you've got this inner membrane folded up inside in there. So the inner membrane space is in between that inner and outer membrane. You've got the mitochondrial matrix that is enclosed by an inner matrix. It's an enclosed inner matrix that's going to have your enzymes and DNA in there, and it's highly folded in there. So all of these folds in there form your cristae in there. When we look at the chloroplasts that we use for photosynthesis, these are a member of the plastids organelles. They store starch, so they're an amyloplast. Amylose is starch. And the chloroplasts will store pigment. Inside, instead of having the membranes, you've got a bunch of interconnected membrane sacs. So where you have the cristae in your mitochondria, in the chloroplast you get these thylakoids. And the granum are the stacks of thylakoids in there, and then the stroma is the fluid outside the thylakoids. And it's going to contain DNA, ribosomes, and enzymes. So in here, this is showing a chloroplast, and you've got these stacks in here, the stacks of thylakoids making up the granum. Another type we have are the peroxisomes. And these are going to help produce hydrogen peroxide in there. So some of it can be used to have oxygen for breaking down fatty acids. When you break down fatty acids, they have to be oxidized, and so it means you have to have oxygen to do that. It also helps to detoxify alcohol in the liver. What else might a cell use it for? What would you use hydrogen peroxide for? To clean. So what you're hoping to do is kill bacteria with it. Well, that's what you're hoping to do when you're cleaning it, is kill some bacteria, whether you know it or not. And that's one of those things that we've been told, okay, you put hydrogen peroxide on it, and you actually, the hydrogen peroxide oxidizes the tissue around it and interferes with the healing. So you don't really want to use hydrogen peroxide for wounds, because it damages the tissue around it. It's more of, you can use it to clean the surfaces and counters, try and get out stains, but for your wounds, you don't really want to do that. It does kill some bacteria, and a lot of times people will look at it and they'll go, look, it's foaming, it's working. When you see the foaming in there, that means the bacteria said, oh, you want to oxidize me? Well, guess what? I've got a way of neutralizing it. And them neutralizing that oxidation is what you're seeing with the foaming in there. So in microbiology, we can actually test to see if a bacteria has the enzyme to neutralize oxygen by putting some hydrogen peroxide on them. It's fighting back. Mm-mm. You don't have an open wound in your throat, but it's not really necessary. The idea that you say, oh, I don't want to have bacteria in my mouth, you got bacteria in your mouth and they're not going away. The only time when you don't have them is at birth. And on your way out, you picked up some from mom. Lovely thought there, I know. So it's just you have bacteria in your mouth. You'll never eliminate all of them in there. So, it can be used for brushing your teeth. It does help to whiten things in there. It's not particularly pleasant. I mean, there's a lot of things you could use that aren't nearly as foul as that stuff. I mean, and some people do like that feeling in there, and they'll use it like with baking soda and stuff. And with baking soda, it's slightly abrasive is what it is. You don't want to use it too much to damage your teeth. But baking soda, you can clean all kinds of stuff with baking soda because it's a mild abrasive. Usually you want to rinse it out with water and then put an antibiotic ointment in it. That's not going to feel very good to do. No. I mean, it depends on the type of wound. Like if you get a puncture wound, you rinse it out and you try and, anything that you can see in there that you can kind of use pressure and squeeze out in there. But puncture wounds, it's so deep in that you have a really high chance of getting an infection in there. I mean, I got a bite over the summer, and I immediately had it under the sink, washing it out, squeezing it out, and it was still infected by the next day. And that was even with putting antibiotic cream in it. I mean, it was like go from getting bit to doing that, matter of just a few feet in there. So next time the dog gets to get in the barbecue, I'm not going to try and save his blind little butt. So glyoxysomes, these are specialized in fat storing tissues of plant seeds, and they help convert fat to sugar to feed the seedling. So when you eat nuts and seeds, they are high in fat content. That fat in there is to feed the new little baby plant. That's why the plant made it. We just happen to like it in there. That fat's not particularly bad for you. Makes nice peanut butter. So your cytoskeleton, this is a fiber network in the cytoplasm. This helps to organize the structures and activities in the cell. It's going to provide mechanical support for the shape. So your cells, while they look like they're just little jelly blobs, they're not. They're actually held in a specific shape like that. It can interact with other motor proteins to give motility, so it will interact with your cilia and flagella. It provides transport pathways inside the cell, so if you get rid of something, you don't just send it and say randomly, I'm rolling it over there. You put it on the pathway and it acts kind of like train tracks to haul things out. It will move plasma to do the phagocytosis, so when you're going to engulf something, it helps to allow you to do that in there. It also helps to regulate biochemical activities in response to mechanical stimulation in there. So if you poke at a cell, certain cells are going to react to that. and it will allow that to happen in there. Just want to see. This is a fairly long chapter. You guys want to take a break for a couple minutes in there before going on to more? I know it kind of gets to a point where you stop absorbing things. Okay, so we're looking at this point, what we started to talk about with the cytoskeleton. And your cytoskeleton is going to be made up of different types of fibers. Okay, so we're looking at this point, what we started to talk about with the cytoskeleton. And your cytoskeleton is going to be made up of different types of fibers. The first one is the microtubules. These are the thickest fiber, and they're made of the protein tubulin. These are going to help to give shape. They're also going to provide support to the cell. They're going to provide those tracks for the organelles to move on. So we talked about that they put them on the train tracks and send them on their way. This is what is going to be your train tracks in there. And it will help to guide secretory vesicles to where they need to go. Along with doing that, they're also involved with making a couple of other structures. We've got the centrosome that is going to be in animal cells, which is going to be a region where the microtubules are going to grow out of. So when you go to divide your cell, you're going to have these microtubules that are going to help stretch across, and the centrosome is going to be where they grow out of. The centriole is within the centrosome, and it's going to be 9 sets of triplet microtubules in a ring, and these are going to replicate before an animal cell divides. However, we do know that they are not absolutely necessary for cell division because they don't have these in plants or yeast cells. The centriole is one of those structures that we don't completely understand yet. So in this model that we have here of cell division, these are going to be your centrosomes here, where your centrioles are going to come out of. And you can start to see where the little fibers are coming out of there. And then in this one that's further along in the process, you can see how they stretch all the way across the cell, stretching down. Now I'll go ahead and pass this around. So in here you can see where there are those sets of 3 in there. So the microtubules are also going to help to make up your cilia and flagella, and they will be just extensions that project out from some cells to propel them. We'll go into more details of those a little bit more here. The flagella are going to have an undulating type of motion. It's more of a swimming-like motion in there. And what is significant about that is it's going to move in the same direction as its axis. So you have a cell here. With its flagella. The axis runs this direction, and the cell also swims this direction. So in a eukaryotic cell, they do more of a swimming type of motion. It is a little bit different type of motion in a prokaryotic cell. It tends to just spin around like a motor in there. Your cilia are going to work more like oars. So the cilia, they go back and forth like this. So they stick out this direction, but their movement is this direction. So the movement is going to be perpendicular to it in there. So your primary cilia are going to act like receiving antennae and help to transmit molecular signals in there. They seem to be important in brain function and embryological development. So they're going to act more like feelers in there, that when something touches it, it can transmit a signal. It's kind of like when you have hair on your arms and you come along and you get close to something and you go, I can feel it, you're getting close to me, you're in my space, you're touching my arm hairs in there. It's the same thing with the cilia. They're going to be in what we call a 9+2 pattern in there that you're going to see in nearly all of the eukaryotic flagella and motile cilia in there. It means you've got 9 doublets of microtubules arranged in a ring with 2 single microtubules in the center. And this picture here shows it where you've got a doublet, a doublet, a doublet, a doublet, same thing all the way around, and then you've got 2 in the center. In your non-motile primary cilia, you're going to have 9 doublets of microtubules and they lack the central pair. So in the non-motile ones, you just don't have the middle in there. So where they are going to attach to the cell, you're going to have a basal body that's going to be an anchoring point in there. So it's kind of like having a little attachment point on there that will hold it on. The dyneins are protruding motor proteins that are going to go along the length of the cilia and flagella. These proteins are going to be what is responsible for the bending movements. So here you can see that it's kind of sliding back and forth, that they start out lined up in there. So because they slide back and forth like this, it allows them to bend side to side. And so this one looks like a sperm. That's what lets the sperm tail go side to side and swim. So it does take ATP or energy to be able to do that in there. So this is just showing them going from being straight, it kind of slides down and it allows it to bend. And then this is demonstrating again the wave-like motion in there. So your microfilaments are the next one. These are solid rods made of actin, so they're made of a different protein in here. You've got a globular protein in twisted chains. So this one is going to be more like lumps that get twisted up in here. This also helps to support cell shape. It's going to give a cortex and cytoplasmic layer for a gel consistency. It's going to help to bear tension forces. So if you're going to engulf something and you're a cell that's going to wrap around and engulf it, you're going to have some tension in that area where you wrap around. So if I'm going to be a big cell here that's going to wrap around and engulf that sperm, there's some tension in this area here. And so we're going to have these additional fibers in there, the microfilaments, to help make this stronger so that this doesn't get lobbed off in the process of doing that. They're going to make up the core of microvilli to increase surface area. They're going to help with motility in there. With your muscle fibers, this is going to interact with myosin, another protein in there, to allow your muscle fibers to contract. The amoeboid movement by extending the pseudopodia in here, so giving this extra strength to be able to slide along and do that. Also in cytoplasmic streaming, so the circular flow of the cytoplasm within the cells. So your cytoplasm inside the cell is going to flow around in there just like you have blood flowing around inside your body. This helps keep it moving. So this is just showing it here, being involved with muscle cells, that you've got the actin that's going to interact with your myosin in there, where you've got making a cortex in there and an inner area. The amoeboid movement showing this kind of moving around in here for cytoplasmic streaming in there. And we've got one more type of filament in there, your intermediate filaments. These are larger than the microfilaments in there. They're also specialized to bear tension in there. These will persist after the cells die. So it can help leave the framework of a cell behind in there. They make up the nuclear lamina lining the nuclear envelope and help give the shape and framework. So you can see there's some overlap in what the different fibers are used for in there. You may see that you have that bearing tension is handled by multiple of the fibers. They're not mutually exclusive in their jobs. So when we look at extracellular components, these are going to be things that are outside the cell itself, outside the cell membrane. First thing is the cell wall. You've got an extracellular cell wall in plants. This is going to help to protect it, helps to maintain the shape, helps to prevent excess water intake. So inside you don't need to have cell walls because you've got a skeletal system that helps hold your shape, as do all other animals. Plants out there, you look at that tree out there, it doesn't have any bones, yet it stands up nice and tall in there. Cell walls are what allow that to happen in there. So it helps hold the plant up against gravity. You'll see some in prokaryotes, some fungi, and some protists in there. A cell wall is going to be thicker than the plasma membrane, and it's going to have microfibrils made of cellulose in there that are embedded in the matrix of other polysaccharides and proteins in there. Various compositions will be seen in different cell types in there and different species. This is what gives plant matter the fiber. So when you eat plants, you're going to increase fiber in your diet. You don't necessarily have to eat grass to consume some cellulose. You're going to get a little bit of cellulose in any plant matter that you eat because of the cell walls in there. So a primary cell wall is going to be what a young plant cell first secretes. It's relatively thin and flexible in there. The microtubules in the cortex help to guide the cellulose in there and orient the cellulose to deposit and affect the growth pattern in there. So when you look at a young plant, if you look at maybe you grow a garden and you've got a little bean that starts coming up out of its seed in there, you'll notice they're really soft and flexible. If you go to transplant them too early, they just fall over when you water them, or if you hit them hard with the garden hose, they just tip right over in there. The primary cell wall is not strong. The middle lamella is going to be between the primary walls of the adjacent cells. It's a thin layer of sticky polysaccharide, pectin and glues, made of pectin, and it helps to glue the cell walls together. We use this pectin in there when you buy pectin in the grocery store. What they've done is they have isolated this pectin in there to be able to use that. And it helps food stick together in there where it's gluing cells together in there. Your secondary cell wall is going to be between the plasma membrane and the primary cell wall in a mature cell. This is going to be the part that is stronger. In some cells, it's just going to be the primary cell wall that hardens. So we need to be able to have these cells be able to communicate with each other, and so we've got channels called the plasmodesmata that are going to be between the cell walls. And this is something you're going to see in all plants in there. So this illustration here shows it nicely, where you've got cell walls next to each other, and these little channels in between are the plasmodesmata. So we're also going to have an extracellular matrix in there, and this is going to be common in animal cells. It's going to be made up of glycoprotein. The most common one is collagen, and it's going to help to form strong fibers outside the cell. So this picture here is showing some where you've got some nice strong collagen fibers outside. The proteoglycan is a small core protein that's going to be linked with a lot of the carbohydrate chains in there. And it's going to form a network where the collagen is embedded. So the proteoglycans around the outside are these green fibers that are wrapped around here in there to help hold the collagen in place. And then the fibronectin is going to bind the surface cell proteins called integrins into the plasma membrane. So you've got the fibronectins right here that you can see. And then the integrins, like their name sounds like, integrates, is going to span the plasma membrane, and it's going to bind on the cytoplasmic side to proteins in there, and that's going to help to transmit messages from the inside and the outside of the cell. So even though you've got a membrane and things are separated in there, the cells need to be able to communicate with their outside world, and this is how they're going to do that. So the cells need to be able to talk to their neighbors too. And that's what we have the intercellular junctions for. So we've got the plasmodesmata that we talked about in the plants in there that are your perforating channels. Cytosol is able to pass through that and connect to adjacent cells. In animal cells, you'll see some tight junctions in there where you're going to have this in your skin. They're going to be bound by specific proteins in here. This is what helps to make your skin waterproof in there. So you can put your hand in water and the water doesn't all flow into your skin. It can't get in between those cells because of the tight junctions. The desmosomes, these are going to attach muscle cells to each other. And these are going to have intermediate filaments of keratin that are going to help hold them together. And then gap junctions are another type of one of these. They're a cytoplasmic channel. This is what you're going to have in your heart muscle. This is what allows all of those muscle cells to work together so that your heart can beat together as an effective pump, not having every muscle beating at its own rhythm in there. So one thing to keep in mind with the cells is we've got all of these parts that we look at, but the cell itself, its function is much greater than just the sum of its parts. When all of those parts work together, you get a much bigger function out of it than just what you see with the individual things in there. So the next is Unit 7, and I'm going to go ahead and stop